is CORDIScovery. Hello and welcome to this episode of CORDIScovery with me, Abigail Acton. Things are moving in the world of prostate cancer treatment. Findings just announced from the University of Oxford show that a special type of fluorescent dye might help surgeons find areas of cancerous tissue not picked up by the naked eye or other clinical methods. A new dimension in precision surgery is how it's been described by Professor Freddie Hamdi, the lead researcher. Meanwhile, a new, cheap and simple testing method using saliva could help catch the disease earlier by identifying the genetic factors which make men more likely to develop the condition. A leap forward in diagnostics and in treatment would be very welcome. Prostate cancer is the second most frequent malignancy in men. In 2020 alone, around 1.5 million new cases and almost 500,000 deaths were registered worldwide. The chances of survival depend on the timing of the diagnosis. Almost half of those diagnosed will survive their prostate cancer for 5 years or more if it is caught at an early stage. Here to talk us through about some of the latest innovations brought about through the support of EU science research funding are Jan Tkac, the founder and chief scientific officer at Glycanostics in Slovakia. The company has developed an innovative diagnostic test for cancer based on novel biomarkers. Jan is also keen to explore how different sports relate to levels of endorphin release. Hello, Jan.
Hello. Thank you very much for the invitation.
It's lovely to have you here. Harald Mischak is the scientific officer at Mosaiques Diagnostics Therapeutics AG, Germany. He is focused on understanding the underlying molecular structures of certain types of cancers to identify the most appropriate therapeutic targets and drugs. Welcome.
Hello. Nice to be here.
Excellent. Joaquim Mateo, who is group leader at the Translational Research Group in Prostate Cancer at the Vall d'Hebron Institute of Oncology in Barcelona, is also medical oncologist at the Vall d'Hebron University Hospital. Joaquim is especially interested in the development of novel forms of precision medicine. Hello, Joaquim.
Hello, Abigail, and to everyone listening.
Yeah, and I'm going to start with you, if I may. ProScan developed an innovative glycan-based early diagnostic method for prostate cancer. So can you explain what glycans are and why are they interesting the scientific community at the moment?
Okay, thank you very much. So glycans are complex carbohydrates attached to proteins or lipids. And actually, glycans are present on every single cell in our body. And actually, it's estimated that up to 70% of all proteins in our body is glycosylated. So glycans are able to provide really very useful information about physiological status of the cell. So this is one way how glycans can be used. And the other one is the fact that glycans can also provide information about development of some diseases or progression of some diseases, including cancer. And actually, this is why we are using glycans as cancer biomarkers.
Okay. All right, then. They give an accurate picture of the status of what's going on. So the ProScan project was hosted by your firm, Glycanostics. So what's novel about your approach? Because I know scientists are interested in glycans. What are you doing differently?
Okay, so actually, what we are doing, we are looking at specific glycans on specific proteins. For example, in case we would like to diagnose prostate cancer, we are looking on differences in glycan composition on one protein. In our case, it's PSA, it's prostate-specific antigen. So how does it work? So actually, we are working in serum samples. Serum sample is a very complex sample containing hundreds of different proteins or glycoproteins, DNA, and other molecules. And actually, we are using magnetic particles modified by antibodies. And actually, those antibodies are able to selectively interact only with PSA molecule in this complex sample. So then these PSA molecules actually also attach to magnetic particles. Then we are using really small magnets for separation of magnetic particles, and everything is washed away. And in the final step, we are using other proteins called lectins, and those lectins are able to selectively interact with some glycan structures. And actually, we are using information about differences in glycan structure for diagnosing of many diseases. So on one side, we are using PSA, which is tissue specific. And on the other side, we are using analysis of glycans, which are cancer specific. So by combining, we can then accurately diagnose, for example, prostate cancer.
Okay, this is fantastic. So basically, the magnetic particles attached to the glycans that you're interested in, and you can extract those simply by extracting the magnetic particles, and they follow. That's fabulous. And is it a complex test to run? Do you have to use new equipment, specialized laboratories, or? No,
actually, assay is very simple. So it works in ELISA format. It's very complicated named. So it's enzyme-linked immunosorbent assay. But actually, this assay is very simple. So it runs only using ordinary equipment. We call it ELISA reader. Actually, this reader is in many different laboratories worldwide, and actually it's also in clinical laboratories. So there is no need to use some special infrastructure. What is really needed, it's to use only ELISA reader. And actually, the assay using magnetic beads is very simple. So magnetic beads allows us to work with, for example, PSA protein attached to magnetic beads. We don't need, for example, to release PSA from the magnetic beads and then to analyze this PSA separately. Everything can be done on the surface of magnetic beads. So assay is very simple. It can be done in short time. And also we can then use really a small amount of sample. For example, few drops of blood serum is really sufficient for the analysis.
Yes, obviously, I mean, one of the problems with cancer generally, but with prostate cancer, is the necessity for biopsies, which can be, tricky to achieve. They're expensive, and there can be side effects and so on. And I believe that your technique greatly reduced the need for that sort of tissue.
Yeah. So actually, we are working with serum samples. So this is this means that our approach is early stage cancer diagnostics. And this is really needed because, for example, urologist needs to manage a patient properly, for example, to suggest if this patient needs to undergo a biopsy or no. And actually, our test can provide second opinion to guide a urologist if patient needs to undergo a biopsy. And actually, in last clinical validation study, we show that our test is the best one from all serological tests which are currently available on the market. And actually, we could significantly reduce number of biopsies using our test.
Excellent, which is stress and money and time, which is obviously crucial in the case of diagnosis for cancer. And how is the system working in practice? So is your system of analysis actually in place in certain health centers and environments and hospitals? Or when could people expect to be able to benefit from this innovation?
So actually, when something works in ELISA format, then it's very easy to transfer this technology also on highly automatic machines of industry leaders. And actually, in our case, since we are using magnetic particles in our assay, this transfer is more convenient for big diagnostic or pharma companies at later stage, because all those highly automatic machines actually involve the use of magnetic particles. So this is really beneficial in our case, and currently we are in Slovakia running testing. So actually we have some agreement with urologists in Slovakia, so patients can pay for this kind of service, and urologists can provide really very useful information on how to manage the patient.
Okay, that sounds wonderful. You must be very proud of all the work that you've achieved so far.
Yeah, it was really hard work, yes, but it's good that this test can be actually used. So we started in Slovakia, and then we would like to extend use of this test in other countries as well.
Yeah, of course, absolutely. How long did it take you to develop this system? Because it must have been a long time in the pipeline.
Yeah, so actually it started with the ERC grant. It was in 2012. Then in 2017 we established a company. But then it took quite significant time, for example, to get finances and investment to fully develop the product. So from the start of the company, it's almost 7 years right now. So it was quite a long journey, but
yeah,
it was a really nice experience.
Yeah, but now finally you're actually going to see the benefit when it comes out. I think that's wonderful. Thank you so much. That was a very interesting explanation and beautifully set out. Thank you for that.
Thank you.
Thank you, Jan. I'm going to turn to Harald, please. Harold, as drugs act on a molecular level, the molecular differences between patients means that while a drug can benefit some, it might not be effective for all. CAR-Pro-Treat wanted to unravel this for better diagnosis and treatment. So, Harald, like Jan, your project also considered less invasive ways of diagnosis, in this case, non-invasive urinary biomarkers that enable early detection of prostate cancer. Can you tell us more about this form of analysis first?
Yeah, that was developed out of a project to basically identify different diseases based on urinary peptides. You have to consider that in general, all smaller proteins and peptides are present in the end in urine. So whatever is in the circulation will be filtered, will be in urine. And urine has the main advantage from a technical point of view that it's quite stable. The urine has been stored in the bladder for several hours at 37 degrees. So whatever we can detect in there is stable and can therefore also be shipped. There are no sampling issues.
Okay.
It works quite well for kidney cardiovascular disease. And then we also applied it for prostate cancer, among other cancers, and found there are many specific proteins and peptides in urine that indicate the presence of cancer.
Okay, so I know that your project gathered molecular proteomics data from thousands of prostate cancer patients. So what did that enable your team to do?
To identify which proteins and peptides are significantly associated with cancer, also with prostate cancer in this way, which proteins are indicative of aggressive cancer. You may consider that most prostate cancer patients do not die from the cancer, but with the cancer. That is a big issue. So in most cases, I would also argue it would be better for the patients not even knowing that they have cancer if the cancer is insignificant anyway. Anyway, these proteins in the end are to some degree also responsible for the cancer. So therefore they form, if you want to say, that the molecular basis of the cancer. Knowing these proteins means I know where exactly to interfere. And using all this information theoretically on all of the proteins that are changed, I can predict which molecules, drugs or whatever I can use to revert these changes, basically.
Okay. So it's like a comprehensive profile of the particular cancer that that patient has. Gives you all the, like a real thorough painting, like a portrait of a particular cancer.
Yes. Also, cancer is not a cancer, so to speak. You know, cancer is quite heterogeneous, and this microheterogeneity in cancer really is the big problem why typically a cancer cannot be cured easily.
Right. And I know that your project was really looking at a multi-omics approach. Can you explain what a multi-omics approach is? And I believe it's quite innovative.
There are different so-called omics techniques: proteomics, investigating proteins; transcriptomics, investigating the DNA transcripts that encode the proteins; genomics, investigating the DNA, and so on. All of these have in common that they investigate thousands of different molecules. And some of these are changed in disease, and together they, of course, are able to, let's say, give a more comprehensive picture of the specific disease.
Right. Okay. So it's like a holistic approach.
Yeah.
Yeah. And then, of course, you end up with a huge amount of data. So
yes,
there must have been computational work in your project as well.
Yes, there is plenty of computational work involved, obviously. Plenty of experts, and certainly the work has profited a lot from collaboration with multiple different scientists in the field of, let's say, bioinformatics, systems medicine, and so on, where I come from as well. We also have benefited basically from our experience in molecular diagnostics, where we run into the same problem.
Right. Well, I'd like to talk about the molecular diagnostics now, actually. So, I mean, as we've just said, cancers are very complex structures and notoriously hard to treat. So I know that one of the things you were interested in was reverse engineering what was going on actually to cause the cancer growth. Could you tell me a little bit more about what you meant by reverse engineering in this concept?
In principle, as I said, the cancer, basically any kind of disease, is determined on the molecular level. You have specific molecular changes associated with disease. Some of these are, in fact, decisive and decide on the direction of disease. If you know these changes, you can use small molecular weight compounds or also antisense RNA, antiproteins, whatever, to revert this change. You bring the molecular signature of the cell or the accumulation of cells back to where it was before, and this would imply that the disease in this case the tumor, is also reverted. And this is what you would call the reverse engineering. We know about a lot of drugs and compounds, how they interfere or how they change expression of certain proteins. And then we just overlay these different patterns to predict which of the drugs may be most useful in a specific type of cancer or for a specific patient.
Perfect. Thank you very much. That's a very clear explanation. I'm appreciating both of these fantastic explanations. So, so building on these findings, you were looking for potential drug candidates. Where are you up to with this? What did all of this analysis and complex computer work as well show you? Where does it leave us?
Yeah, well, but now we have 7 drug candidates shortlisted, and these we now need to investigate in more detail. We need to demonstrate where we know from some of them by now that they in fact revert the tumor phenotype in cell lines in vitro, as in the tissue culture. This is where it stops, because every approach beyond is quite expensive and carries a huge regulatory burden. And here, unfortunately, I have to say, we have to try to find funding.
Yeah. Yeah, I mean, it must be quite frustrating when you know you're sitting on something that is actually potentially really, going to save lives and turn situations around, and presumably not just for prostate cancer, but perhaps could be applied to other cancers as well.
Yeah, you're right. It is frustrating. We have to find funding from private venture capital companies, which is what we are doing. And all the patient organizations by now support us and try to find the funding, because in the end, of course, the patients would benefit from an improved drug.
Yeah, no, potentially life-saving. Absolutely.
Yes.
Okay, thank you so much for explaining that so clearly. Does anyone have any comments? Yes, please. Joachim.
Hi, I wanted to ask my colleagues, I mean, first of all, congratulations for your work. It's really amazing. But I wanted to ask Harald if, beyond the applications for diagnostics, have you ever tested whether this urine test could actually detect a relapse of the disease on a patient that has already had surgery for their prostate tumor?
We have not investigated this in detail. I would assume, yes, we tested this in a slightly different context in bladder cancer, where the tumor is removed, but as you know, frequently these patients develop novel or relapse of the tumor. And there we tested it and we found that, in fact, yes, we can detect the relapse of the tumor. Of course, it's a matter of size also. So I would say that it's difficult to detect a few tumor cells. But once the tumor reaches a certain size of maybe 1 to 2 millimeters, it's quite easy to detect using this technology.
Fantastic. That's excellent. That's really, really good. It's almost like a thermometer, sort of just, keeping an eye without being invasive. And again, presumably also, as you said, the urine stores well, it's easy to access, it's easy to use. Brilliant. Thank you.
The one advantage certainly is, I want to point this out as well, that we now have data from close to 90,000 patients with different diseases and controls. So this helps to be quite certain about the statistics behind it.
That's an awful lot of patients. Gosh. Mm-hmm. Where were they all taken? What database is this? Is this some sort of collective database, obviously?
Yeah, there's a collection of all, well, we were involved in many European projects. It's a collection of all of these samples and data.
Okay. Fabulous. I mean, that's very rich resource, very useful. Super. Okay, thank you so much. Joachim, I'm going to turn to you now. The AR-DDR project looked at the exchange between 2 key pathways involved in prostate cancer development. So you're interested in treatment. We've been looking at diagnostics. Now we're turning to treatment. You want to optimize precise therapeutic options. So when you say prostate cancer, we think of one single disease, but as Jan was explaining as well, that's not quite accurate, Joachim. Can you tell us more about the condition, please?
Yes, I think that we have just heard from our colleagues 2 brilliant studies on how to better diagnose and treat initial stages of prostate cancer. Fortunately, most prostate cancer are diagnosed at a time where the disease is confined to the prostate and can be cured. But we know that certain patients develop upfront an advanced prostate cancer condition. So the cancer has spread outside the prostate to other organs. We know very well now that this, what we call localized prostate cancer and what we call metastatic or advanced prostate cancer, are 2 different biological entities, and that the molecular features of the localized or advanced cases are very different. So that's a fair difference that tells us that not all prostate cancer are the same. But when we go to the field where I work on, and maybe I'm a bit biased because I work on the minority of prostate cancers that are very aggressive and had spread outside the organs, we still know that within that patient population, the tumors may be very different to one another, genetic level and many other levels of biological differences. We knew that. What we know now is that these differences can be leveraged to choose the best treatment for each of these patients, because we may be able to predict to a certain extent whether one particular tumor is more likely or less likely to respond to one specific drug. So, until 10 years ago or 15 years ago, we didn't have that many drugs to treat advanced prostate cancer. So in fairness, there was not that much research about how to choose one treatment or another, because there was nothing to choose from, right? But as we have developed more drugs that can slow down or even make the prostate cancer regress in the advanced stages, there is more and more interest in understanding which biomarkers, which biological measures can help us select the best treatment for each individual patient.
Okay, that's fascinating. Well, Harald's project was considering the molecular signature of the tumor itself. And in an earlier episode of CORDIScovery, we considered the interplay between bacteria and the microbiome and response to chemotherapy. Joaquin, tell us about the additional complexity of genetic differences between patients and how that impacts on treatment.
So we know that the prostate cancer is primarily driven by the male hormones, the androgens, okay? And almost all prostate cancers are somehow sensitive to manipulating the androgen pathway in the body. However, beyond that, there are other pathways that help the cancer cell survive that may be different from one patient to another. And some of them may be suitable for therapeutic targeting. It is true that when we conduct research on what we call precision oncology and identifying biomarkers, many times we kind of pick one gene or one protein and then see how it changes the sensitivity of the tumor. But it is true that the biological pathways in a tumor are interconnected. And the overall complexity of cancer is much higher than just targeting one protein. So part of this study and many others that are in the field is in trying to understand how one same genetic alteration or a gene mutation may have different effects when it comes to sensitizing to a drug, depending on the background where they occur.
Okay, that's great. So you were looking, I think, at the presence or the absence of a particular gene known as the ATM gene. We don't need to go into more detail about the name, but what was so interesting about the ATM gene? What were you considering there?
So for the last 10 years, I've been working in testing a family of drugs called PARP inhibitors that were already approved before in certain forms of ovarian and breast cancer. And we actually found out that they were working in certain prostate cancer patients. These prostate cancer patients were the ones that had mutations in a series of genes. Okay, one of them being this ATM gene. Okay, what we realized when conducting clinical trials is that despite all these genes have very similar functions, depending on which one was lost, the response of the tumor was very different. And ATM was very puzzling because we really had a situation where some patients with these mutations did very, very well on the treatment, and some other patients, didn't benefit at all. So we wanted to go back to the laboratory and understand what does this ATM gene does for prostate cancer, and actually whether it's a truly sensitizing biomarker for this family of drugs.
And 10 years is a long time. Do you have some findings for us? How is the work going?
Yeah, actually, we've been working for a bit more than 10 years on developing the drugs. These drugs are now approved and available for prostate cancer patients.
Oh, fantastic. You had the drugs pass the approval process.
Correct. They were approved in 2020, but still there has been a lot of controversy into which are the patients who should get the drug. Because as I was saying, depending on the genetic background of the patient, the response may be more or less important for the patient, or short-lasting or last-lasting, right? So then in the last few years, we have been looking at the impact of this ATM gene in laboratory models of prostate cancer. We have been trying to delete this gene from cancer cells in the laboratory to see how in different tumors, the loss of this gene may infer different levels of sensitization to these drugs.
Right. And data coming out, are you establishing what that looks like, what the loss is showing?
Yes, so we published the findings of this study in 2021. One of the things that we realized is that when this ATM protein is lost, there is another backup protein in the cancer cell that takes over, and sometimes that's enough for the cancer to grow again. With that, we actually went into testing another drug that is targeting this backup pathway. And we realized that for the patients with this mutation, in particular in the ATM gene, combining the two drugs together was much better than just using one or the other. And so research for patients, but actually we demonstrated that in laboratory models. We are now trying to do a subsequent clinical trial testing in patients the combination of these two drugs. Although I have to say it's a bit challenging because both drugs, because they are so similar in the proteins that they target, they also have similar side effects. So we are trying to see how to combine them in a manner that is safe for patients.
Right. Okay, so that's the next stage. Fascinating. That's absolutely brilliant. So you've worked out the mechanism behind it. You've realized that the cancer gets around it by providing a different pathway, and now you're targeting that secondary pathway. I think that's fabulous. Really, really great. Super. Does anyone have any questions for Joachim? Yes, Harald.
Yeah, maybe one question. Did you have similar experience that it gets extremely difficult to test drugs in patients in the end? So what are your experience with talking to the regulators?
Well, that's a very long answer, probably. But yes, I mean, going into clinical trials is a very challenging phase of developing a biomarker or the drug. And one of the main issues for developing trials in the manner we wanted to investigate this is that, running a clinical trial is hard enough. If you have to prospectively define genetic biomarkers for tumors, it makes it even more challenging because you have to first identify those patients who may be candidates for the testing of the drug and then conduct the trial, right? And that, ends up adding layers of complexity. In general, I think that is much more difficult in our current regulatory framework in Europe to run trials to validate biomarkers than just to test the drug. I think we have a very good setup for running clinical trials of new drugs in Europe and testing if they work or if they don't. But when it comes to linking the effect of a drug to a specific biomarker, the testing and the process of generating the sufficient evidence for approval it's more challenging. And I think that we are also now going through a phase in Europe with the approval of the new in vitro diagnostic regulations, what we call the AR-DDR regulation, that it is making it even more challenging for a good reason, right? To ensure the level of stringency in the test of the biomarker, that every time that you test for a biomarker, you only get the exact result, an accurate result for that biomarker, right? But it is also true that it is dificulting a bit the way we conduct biomarkers clinical trials.
Thank you very, very much. I'm very, very grateful to you all because this was a technical subject and I think you've all explained it beautifully. It's been very transparent and very clear, and I'm grateful. I'm also grateful for all the hard work that you're all individually doing and particularly in the face of the challenges that Joachim and Harald have just been talking about, which on top of all the hard work must, after a while, get a bit tiring. So thank you for sticking with it. Much appreciated. Thank you very much.
Thank you.
Thank you. And thank you for listening.
Yes, thank you. Thank you for the interest, Abigail.
Yeah, well, we're all very interested in this. Of course we are. Wonderful. Thank you. Bye-bye.
Bye-bye.
Thank you. Bye.
Bye-bye.
If you've enjoyed this episode of CORDIScovery, follow us on Spotify and Apple Podcasts and check out the podcast homepage on the CORDIS website. Subscribe to make sure the hottest research in EU-funded science isn't passing you by. And if you're enjoying listening, why not spread the word? We've talked about skin patches applied to detect TB and how access to new food sources shaped prehistoric cultures. In our last 37 episodes, there'll be something there to tweak your curiosity. Perhaps you want to know what other EU-funded projects are doing in the field of cancer research. The CORDIS website will give you an insight into the results of projects funded by Horizon 2020 and Horizon Europe that are working in this area. Take a look at our pack of projects called Frontier Research for Cancer. The website has articles and interviews that explore the results of research being conducted in a very broad range of domains and subjects, from voles to volcanoes. There's something there for you. Maybe you're involved in a project or would like to apply for funding. Take a look at what others are doing in your domain. So come and check out the research that's revealing what makes our world tick. We're always happy to hear from you. Drop us a line, editorial@cordis.europa.eu. Until next time.